Hello, my name is Topolsky. I work as a software developer at the Swiss Institute of Bioinformatics, and in this poster I'm going to show the work that has been done in our computational biology group at TH Zurich on VPipe, our viral genomic pipeline, as we are currently applying it to SARS-CoV-2. RNA viruses have characteristics which have impact on the dynamics of their evolution. They have high mutation rates, they have short generation times, and exist in large population sizes. This gives rise to both inter-host and intra-host genetic diversity. Such viruses can exist as a mixture inside a host. Over time, new variants might arise which can carry characteristics which are impacting on the treatment of the patient. That might be resistance to a drug, or as recently discussed in the literature about SARS-CoV-2, that might be a new receptor. Over time, such a variant can be selected for and take over the population up until the point at which its impact can be seen on the host. Traditional sequencing method can only show an overview. They cannot show the diversity which is happening inside the host. A solution to be able to see the diversity and see the rise of new variants much earlier would be using next-generation sequencing. NGS technologies allow for seeing such fine details, but they come with their own shortcomings. The reads are short and the method is prone to error. To address these problems, a new software pipeline was introduced by the SIB in 2017, VPipe. VPipe takes as input the raw reads coming from the sequencing machine and a reference for the virus being studied. It can perform quality control, it does align the reads, it generates consensus sequence and then calls the SNVs against this sequence. It can also generate haplotypes either at the global level or at the local level inside Windows. The code of VPipe is available on GitHub. It now features a new installer for quick and simple installation, and the pipeline relies on SnakeMake. SnakeMake will help install the component of the pipeline, both our own component, which were developed here at the CBG at ETH, and the third-party component that we're using are all available under open source license and are available as packages on Bioconda repository. One example of such component that we have developed is Shura, which is in charge of reconstructing local haplotype and calling the SNV. It works by sorting the reads inside Windows, then per window trying to cluster similar reads together and trying to eliminate the errors which will going to be seen as spread randomly all over and not clustering together. Once these local haplotypes have been generated, calling the SNVs with a high confidence of eliminating the errors is easier. More information about VPipe and how to use it can be found on the wiki. Since the beginning of our research on SARS-CoV-2, we have also released a tutorial to help other people run it on their own datasets, and we have introduced a new installer which helps with a single command to install the software and get it ready to be used. The tutorial will demonstrate it. To facilitate work with downstream analysis of the results, now VPipe produces its variants using standardized VCF files. Another new addition is a visualization interactive report which helps people look at the various SNV, look how they map on the known annotated region of the genome or other interesting regions such as, for example, primers, and can be filtered by quality level. One of the current applications of VPipe is using it to help automate the sequencing of SARS-CoV-2 sequences. Swab tests which have been detected positive by the Violi laboratory in Switzerland have been obtained by the genomic facility in Basel. They perform sequencing analysis using an Illumina MySeq, and the raw data are uploaded into their OpenBIS repository. VPipe takes care of automatically downloading the raw reads from OpenBIS, performing the analysis, also doing quality checks and making reports to assess the quality of the results. The consensus sequence and then made available to the computational evolution group for downstream phylogenetic analysis and released through GISAID, enabling Nextstrain to draw a phylogenetic tree of the sequences found in Switzerland. These sequences have now joined the amount of information which is available from the Swiss National COVID Science Task Force to help getting a better picture of the evolution of the disease. And with that, I would encourage you to come visit our website. You will find a mailing list where you can get subscribed to get information about new releases and other such announcements. You can find email address to which to ask questions. You will find tutorials and much more information about vPipe. I want to thank all the developers on our team who have contributed to make vPipe. I want also to thank all the external collaborators with whom we had opportunity to work with and to develop our current technology. Thanks also to the past members who have also helped in developing the previous version of the pipeline. Thank you for listening to this poster presentation.